entrambi e comunque la voce non è che ho visto che è abbastanza...
No, la voce c'è. La voce c'è sicuramente. Magari guardando di qua mi sentono. Se parlo così mi sentono? Sì, si sente? Ok. Okay, I think we're all right. 4, 8, 9, 10. Yes, it's all of us. So, I divided the course of this week in 3 parts. Very general part about what microbes are and a few things I think you need to know in order to do and think about hydrogen microbiology. Then tomorrow we're going to spend time discussing mainly metabolism, especially how it relates to hydrogen and other compounds that are common in reservoir or they're common in geological settings and how things interact with each other. Tomorrow we'll discuss a bit what's the relationship between microbes and thermodynamic, what's the relationship between microbes and rates of reaction in the environment. I think that will be relevant for a lot of you that are working, for example, on modeling underground hydrogen storage, but not only. And then on the third day, we're going to discuss the microbiology of hydrogen in general, not just in underground storage sites, but in general of hydrogen. Thursday morning, couple of classes, one on culture dependent, one on culture independent techniques, on how to sample things. It's not unlikely that you might end up in the field with microbiologists that are sampling for microbial diversity in the context of underground hydrogen storage. At least knowing what they're doing or why they're doing it in a certain way, it might be useful. This might help you also to collaborate with microbiologists in the future, knowing what they need and what they care about. And then in the afternoon on Thursday, we have a few use cases that we're going to present from a variety of different hydrogen-related things we've been doing. I have to say there has been a challenge to try to condense and simplify microbiology in 3 days. So for a lot of things, I had to simplify, okay? And a lot of things I'll tell you are general rules or general features, but keep in mind that there are always exceptions. There are exceptions of many types in microbiology as in any discipline. So if you get curious about something, you might always read more later. I left a list of reading papers or reading resources at the end. I'll share with you the slides so you can go on and find some of the things. But most importantly, since there's a lot of things I want to tell you, but there are very few things that I want you to be I want you to have very, very clear. I'm going to stress those multiple times. Please ask questions. Please stop me. Please, anything. Doesn't need to be exactly what I'm talking about. If this clicks with you with something you are doing, and you see a connection there, or you have a question about how it relates to what you're doing, stop me and discuss. So it gets better for me and for you if you stop me, if you don't understand something is not clear, or something sounds different where you come from. I'll give you just an example. When I started many years ago to collaborate a lot with geologists and volcanologists, I joined the field expedition in Costa Rica, and we spent 2 weeks and a half, almost 3 weeks in the field with an international group of geochemists, microbiologists, noble gas geochemists, volcanologists, and so on. And we were discussing about volatiles a lot. Now, if I say volatiles, what do you think about? What's a volatile for you? Louder, please. Water vapor. Anybody else? If I say a volatile compound, what do you think about? Ethanol or low hydrocarbon things that are in vapor form at room temperature, right? Anybody else? Do you all agree is either water vapor or organic carbon? Well, when you work with volcanologists, volatiles are a lot of other things, including chloride, iodine, sulfur, ammonia, all the things that for a biologist are usually either solid or ions. When you are at high temperature, high pressure, they're volatiles. They're not behaving like a normal ion in solution. They escape as gas. So depending on the reference framework and your background, common words might mean something different when you come from. That's why whatever I'm talking about, if I say something that doesn't resound for you, or it sounds strange, or means a different things where you're coming from academically, let's stop and discuss. It gets better for you, it gets also better for me. Okay, because we have very diverse background here. Any question about this? Any comment? No? Okay, so why to study microbiology? We study microbiology for a number of reasons. We started as a human species because of pathogens, because of disease, because people were dying. We had no idea there were microbes in the environment. But the germ theory started to explain some of the disease we were getting through the presence of something that was invisible and passing through air. So we started doing microbiology for survival. But then we discovered that microbiology is actually more important than just pathogenesis and disease, and is actually related to the entire functioning of the planet. And even if microbiology is a relatively new discipline compared to many of the other disciplines you guys are coming from, it became quite relevant in a lot of fields. Right now, we study microbiology to understand the origin of the evolution of life on this planet as a model for basic life principle and how life works. Many basic mechanistic understanding of how life works, they come from microbes, as a model for the search of life somewhere else, because we think that there is life out there, but it's probably microbial. But most importantly for you, we are using it to understand what's the contribution of microbes to the functioning of the planet. And if you are into underground hydrogen storage, you are interested in green energy. If you are interested in green energy, you're probably interested in climate change and global warming. And if you are in this too, you should know microbes very, very well, because they play a huge role in climate change and global warming. And at the same time, because they give us biotechnological application. And the microbiology of hydrogen applied to underground hydrogen storage is a biotechnological application. So several reasons, a couple of them, at least a couple of them are more relevant for you guys, but there's a number of ways and reasons by which we study microbiology. We study them because they dominate what we say is the bio portion of biogeochemical cycles. Now we'll go back to what a biogeochemical cycle is. But the key part is that today, as of today, a lot of the cycling of elements and nutrients on this planet is carried out largely by microbes on the surface of the planet. And a lot of the connection between the bio and the geo in biogeochemistry, we will see, is actually done by microbes. Now, this doesn't only happen today, happened also in the past. This is an image that is called the Earth clock. So you can see it starts at 4.55 billion years ago at 12, and then you go clockwise, you have the Hadean, the Archean, Proterozoic, so you have the different ages eon of our planet. And life started somewhere around 4 million years ago, between the purple and the reddish color. We're not sure yet, 3.8 to 4 billion years ago, that's where life started, more or less. And it started as unicellular organisms that were microbes. And we'll get to a better definition of what a microbe is later. But if you keep going, so you go another billion year, another 1.5, 2.5 billion years ago, 2.4, that's GOE, the Great Oxidation Event, when oxygen started rising on this planet. We're still entirely microbial. You need to keep going all the way to about 700 million years ago to start to get multicellular life. So from 4 billion years ago to 700, 3.3 billion years, this has been a completely microbial planet. Multicellular life started late, complex life came even very late. Mammals are the last, I would say, hour of this clock. Hominids, so the evolution of humans, Homo, 2.5 million years, is a tiny dash, not even a second, you can barely see it. So this has been for a very long time a microbial planet. And so microbes dominate a lot of the functioning of this planet, and they've been doing it for a very, very long time. So if you look at today, about 50% of the oxygen almost that you're breathing right now is produced not by plants, but by microbes. 50% of all the carbon biomass that is produced every hour is produced by microbes. 90% of the recycling of organic carbon, the same organic carbon that then produced coal, natural gas, and petrol, is due to microbes destroying the carbon, recycling the carbon back as CO2, methane, and a number of other inorganic molecules. 100% of nitrogen fixation is biological, divided between human, artificial hydrogen fixation, the Haber-Bosch reaction, I don't know if you're familiar with it, same person that invented the nervine gas, used during World War II, and microbes, which they do it all the time. And they've been doing it in the roots of plants in our fields since 6,000 or 7,000 years ago, when we started actually seeding for the first time and doing agriculture. 80% of methane emission, and we can keep going, and on and on and on and on. So basically, if this planet is still functioning, it's largely because microbes recycle things around. Where do we find it? So the first square on the left is divided in 3 main squares. The green one represents the total organic carbon on this planet, total biomass. The green square is terrestrial ecosystem. About 86% of the total carbon is terrestrial. And if you look on the other side, plants represent the majority. There's only terrestrial, basically. They're not present in other ecosystems. Now, if you stay with the main square, the blue square at the bottom, the small one, it's about only 1% of the total. That's ocean, marine oceanic carbon. As you can see, there's not a lot of organic carbon in the ocean. There's a lot of animals, a lot of biodiversity, but there's not a lot of carbon. And if you look on the right, the blue is majority are protists and animals. Very little fungi, no plants in the ocean. If you look for the other square, the side square in the big one there, 13% of that carbon, total carbon on this planet, is actually in the subsurface. 13% of the total carbon on this planet is below our feet. It's going from the surface of the planet down to depth of several kilometers. We will discuss better how deep it goes and why, because it's very relevant for storing hydrogen underground. But you need to think that the second largest biome on this planet is not on the surface of our planet, is not in the ocean, but is actually underground. And the majority of that underground, as you can see on the right, is made of bacteria and archaea. In other words, by microbes. So microbes present below your feet represent 14% of the total biomass of this planet. Second largest biome on this planet. And typically we look at things like this. People that study microbiology, we look at interconversion of different species of nitrogen between oxic and anoxic ecosystem, between sediments and water and atmosphere, and trying to understand who's making the conversion from one type of nitrogen to the other, and how's nitrogen cycling around. Now, this type of diagrams and reasoning are very similar to what we use in underground hydrogen storage or other type of underground storage to understand who's carrying out possible reaction that might happen within a certain reservoir. So this is one view of it, and this is more on the functioning side. We understand to know how the things are functioning there, who's carrying out things, and then sometimes for biotechnological application. And now, I'm pretty sure everybody knows biotechnological application of microbes, or at least heard a few, right? Does anybody think never heard about any application of microbes? Yeah, CRISPR's technology to edit DNA. That's the immune system of microbes. You heard about microbes used for, I don't know, remediating polluted area and do bioremediation. You might have heard of microbes for enzymes that are used. Microbes are used for, this is probably one of the most influential microbes ever, Thermus aquaticus. Have you ever heard about PCR? Maybe yes, during COVID, PCR, COVID test. PCR is a technique to double and multiply, to make copies of DNA. We used it to count how many viruses we had on us. Well, PCR can be done thanks to one simple enzyme called TAC polymerase. This enzyme makes a copy of DNA under a certain condition, right? Well, that enzyme comes from these microbes here. TAC polymerase stands for Thermus aquaticus polymerase. So a single enzyme from these microbes allowed you and everybody else in the world to do very precise molecular COVID tests during the COVID pandemic. That's an example biotechnological application of a single enzyme of one microbes that contains probably 3,000 enzymes alone. So if you multiply that by the number of microbes on this planet, just think of the possibilities that we can reach technologically, right? So right now, microbes are used for a lot of things. For example, industrial enzymes to break down organic carbon left over of different times in the industry. So you have, for example, leftover from agriculture. You want to break it down to something that can be easily digested and transform into something else. Bioactive compounds. All the antibiotics we use come from microbes originally. Bioremediation, for example, of oil spills, heavy metals, uranium contamination, PCB and polychlorobiphenyl dioxins, and all the chemical as humans we've been releasing in the environment. Microbes can break them down and we can use to clean up the place. We use microbes for crop production. We use microbes for biogas and biofuel production. You heard of biogas, most of you probably. Underground, hydrogen storage. But microbes are also important in classic methane and petrol industry because they're at the base of souring. Have you ever heard of souring of oil wells and methane wells? Yes, no? Souring of oil wells means that your well becomes rich in H2S. And that's a compound that is acidic and you don't want it. And very often the start of this reaction and the production of H2S comes from microbes. And so they're important not just for underground hydrogen storage, but for carbon underground storage, for natural gas production, and so on. Biomining and ore recovery. Microbes are involved in extracting and processing of a lot of different minerals. Bioleaching, yes. Above all, copper. We estimate that 20 to 30% of the copper we use globally every year is actually leached by low-grade ore deposit using microbes. It's not we do it consciously, we select the microbes, we put it there. We just process the ore in a way that provides a very nice environment for microbes to multiply, and their activity changes the redox state of the copper that then becomes soluble, can leach out, and we can recover it. And they've been used since forever for this. We just didn't know there was microbes. Now we are studying microbes to recover, for example, electronic waste. You can do it chemically, but you can do it more sustainably, for example, using biomining microbes that now can be used to recover rare earth elements and metals. And then geoengineering. which is probably the last frontier of microbial planet interaction, because you can think about twinkering with the functioning of the entire planet by interacting with the microbes one way or another. For example, increasing the drawdown of CO2, or shunting biomass somewhere else, or stopping microbes from waking up and breeding things in the Arctic during permafrost melting. Before we go on, since we talk a lot about biochemistry, I want to spend just a couple minutes on telling you more about this. You remember Thermus aquaticus polymerase, the enzyme we use for COVID test? Well, the organism was isolated in 1967. Thomas Brock was a professor in Indiana University. He would drive past Yellowstone every day, and sometimes he would stop and look at pools there of boiling water and see a lot of colors. And he would be left wondering if there was not all life there, what life was doing, why certain color would change. So he started collecting some samples. And a few years later, isolated this organism. And, it was the first organism able to live at 75 degrees. So that was a very nice discovery, but had absolutely no application. It was curiosity driven. So 67, Brock explained, bacteria are able to grow at any temperature which there is liquid water, even in pools which are above the boiling point. About 10 years later, somebody from another university isolated the enzyme from Thermus aquaticus. They were doing structural crystallography. That's a very central enzyme, the enzyme that duplicates DNA. They did the crystal structure. Another 6 or 7 years later, Kari Mullis, that was working at CETOS at the time, was perfecting the technique of duplicating DNA. At the time, the enzyme people were giving to PCR to make the copy of DNA will work at 37 degrees. But at 37 degrees, your DNA is tangled up in the double helix. The way you open DNA is warm it up above 70 degrees. But if you warm it up above 70 degrees, your enzyme breaks down. So what they will do, they will heat up, add the enzyme, let the enzyme work at 37, then heat up again, the enzyme will die, cool, add the enzyme, let the enzyme work, do this into a cycle. Kari Mullis used the thermal stable, the DNA polymerase, the enzyme from an organism that grows at 75 degrees to make a technique in which you add the enzyme once, and then you cycle up and down in temperature, and you make multiple copies all the time. 2 becomes 4, 16, 32, and so on. Now, that innovation allowed PCR to go from a technique that will take days to do in something you can do in 2 hours. 400 million euro a year just for the enzyme per se, hundreds of billions of euro total technology. Now we go from isolating a microbe out of curiosity to one of the biggest business in biotechnology from every day. Basic or applied research at the start of this. So there are different definitions of basic or applied research. I think this is a very interesting debate for you guys overall. I'm not going to spend time to this, but sometimes there's a perception, and you are in an interdisciplinary field, where there are more or less useful things or more or less pure things. Sociologists is just applied psychology, which is applied biology, which is applied chemistry, which is applied physics, and of course, everything is mathematics in the end. Now, things are not that simple because we tend to assume that things go from basic to applied. But we have a great example in history where things went to applied to basic. Can you think about any discipline or anything that went from applied to basic, the other way around? Come on, engineers, I'm counting on you. No? No clue? Not a polite guess? Thermodynamics. We started messing with steam machines long before we had the understanding of how thermodynamic worked. Adiabatic expansion work. This is all used to explain practical phenomena were coming out of the machines, not the other way around. So sometimes we understand the principle and then we find an application. Sometimes we study practical things and then we go back to the principle. So these two things are related. And it's important as people that work in an applied field that you know the relationship between basic and applied science, and you know there is not a one-way street, but it goes both ways. Now, there's a little bit of reading here about this. This is just to say that very often things we study in microbiology for which we have no idea what the application is may turn out to be very important, and vice versa. Things we study in a very empirical way for which we don't have a theory that explains it yet may become important in making new theories that are very general later on. So microbiology is a fairly new. So key message one, microbiology is fairly new. We start with microorganisms in the late 1600s with the first invention of a microscope. Takes a little bit to get things kick-started because you cannot interact with microbes without a microscope, right? That was one of the basic innovations to see microbes. But also, we had to fight a few of the very well eradicated beliefs that were at the time, like spontaneous generation. In spontaneous generation, microbes and insects and other things rise from dead organic matter all the time. Now, a few people, including the Italian Spallanzani, disproved this by getting meat broth, boiling it in a completely sealed tubes, and in this way, the broth will remain clear for days. If the tube was open or broken, then the broth will become cloudy because things will start to grow in it. And then eventually, bot fly and flies and things will be inside. But this effectively demonstrated that you need life to create a life. There's no spontaneous generation there. Life doesn't rise out of inorganic things, at least now. Then, in 1880, we have the germ theory and the first vaccines. Things start to pop up really in the last 120, 150 years. So microbiology is a relatively very young field. And a lot of big innovation, they happen in the last 30 to 50 years. The majority of big innovation, I would say, is probably the last 50 years. And we'll go through it a little bit because I think they're very relevant for the hydrogen. So first things to know, microbiology is a relatively new field, okay? Second things to know, as I was saying before, we are in a microbial world. Now, this is not just a way of saying there are more microbial cells on this planet right now at any given time than stars in the entire universe. We think we have about 10 to the 29 to 10 to the 30 to the power of 30 microbial cell at any moment on this planet. Those are gigantic numbers. And viruses are even more, and we'll see that. So generally speaking, you need to know that if you are within the boundary of life, and we will define what these are, there are microbes there. The question is, you will win the Nobel today if you can find a place on this planet where you are within the boundary of life, but you can demonstrate the absence of life. Because right now, going to a place and finding life is not a surprise anymore. We expect to find life everywhere. You have certain conditions that we will discuss later. And microbes are by far the most abundant life form on this planet. Microorganisms by orders of magnitude above. So 10 to the 29 to 10 to the 30 is a huge number. We think stars in the galaxy are 20 to the 24. So we are 6 or 7 order of magnitude higher microbes on this planet. Now, there are a few things that I think that are also important for those of you doing the modeling. When we say microorganisms, we mean generally unicellular, so a single cell, generally small organism, okay? But small is how small. So there's a big range here because we move from 700 micrometer, so it's close to 1 millimeter in thickness. That's an exception. It's the end member. It's a giant microbe called Thiomargherita namibiensis, all the way to things that are 0.2 micrometer, so 200 nanometers. Those are fully functioning cells. We are not talking about small parasites yet that can attach and do strange things. We are not into the virus. We are still into the microbes. So organisms that are able to do metabolism, to replicate, often to move around and interact with their environment. Now, 200 nanometer is very, very small. And so you have the full range. If you want to know the average in the environment is about 1 micron or slightly less. So your average size in the subsurface, in the environment, will be, I would say, between 0.7 and 1 micron. So microbes are a lot, and microbes are generally very small. But there are exceptions. There's a big range of this. This is an example. Now, this is more useful for you than right now, for later on. But you can see a range of microbes going from the 750 micrometer in size to the tiny one, okay? With a few names, a few metabolisms they can do will make more sense tomorrow. But this is more for your reference. Keep in mind the average size, okay? 1 micrometer or less on average. Size matters for microbes. And we have a upper size and a lower size. The lower size is limited by the amount of machinery you can pack in a very, very small volume, right? You get so small that you cannot fit everything you need to survive. It's like an apartment. If you want to have a bathroom, a kitchen, a bed, a table, there's only, you can go as small as up to a point. After a while, you cannot fit all the equipment, no matter how creative you become. The bigger size, instead, is almost trivial for many of you, is area volume ratio. There's not you need to exchange things with the outside. If you become too big, your area is not big enough compared to your volume. So when things get very big, usually they start to become multicellular. So they split, they aggregate, so they can be big and do more things, but every individual cell maintains a certain surface to volume ratio. Size of microbes, abundance of microbes, are very, very important parameters when you think about their activity everywhere and their mobility in your porous system, for example, and so on. There's an interesting, how many of you know what's a scaling law in physics or mathematics? So scaling law are those laws that when you change in scale from one way to the other, they maintain a certain mathematical properties. There's a scaling law in biology, which is the size to abundance scaling law. So basically it tells you this is a log of, this is in the ocean, for example, those are cells per liter of seawater, and that's the size of organism. And you can see that the bigger organisms are much less abundant than the higher one. You can think it all the way to whale. It's going to be almost a straight line. How many whales you have per liter of ocean? 0.00000, because they're huge, right? And the smaller you get, the more of these you can have in 1 liter, until we have 10 to the 8, 10 to the 9, or 10 to the 10 viruses, which are the smallest one for a liter of seawater. So every time you go out swimming and you drink a little bit of seawater, you're drinking millions to tens of millions to billions of bacteria and viruses, most of which are completely harmless to you, but you're still ingesting all that. Now, you're also ingesting a few zooplankton, copepods, and tiny, few tens to hundreds of micrometer tiny shrimps. But these are just 1 to 2 per liter, 10 per liter, right? Because of this scaling law. So think that the smaller the things you are interacting with, the higher their abundance is in wherever you're thinking. This is not just for seawater, by the way. This applies to grams of soil, kilograms of rocks, volume of air, because there are microbes in the air everywhere. And the smaller they are, the higher their abundance probably. So this is a very universal law that we see anywhere. Now, when we say microorganism, this is just a property of being small. Okay? When you're organism, you're talking about organisms that are micrometric in size. We're not implying anything about what type they are, okay? Microorganisms can have inside all 3 domains of life. And we'll see the 3 domains of life in a second. Generally speaking, though, a lot of people, when they say microorganisms, they're only meaning 2 types of the 3 domains of life. So you are eukaryotes. Eukaryotes means you have a true nuclei inside your cell. You have a specialized membrane that contains the DNA. These guys here, bacteria and archaea, which are the other 2 domains, they're called prokaryotes. In this case, means they don't have a complete nuclei. They don't have a special place where they keep their DNA. Their DNA is out in the middle of the cell. It's like having, a highly hierarchical classified office environment where the boss is sitting in a special cubicle, or having the boss sitting across everybody. Microbes does not imply any of this, but most people, when they say microbes, they actually mean prokaryotes, things without a nucleus, things that are relatively simple in cell organization, okay? And you can see that these prokaryotes, this simple cell, they appear to be, at least superficially, like a small bag that contains fluids and DNA. There are no specialized structure. Of course, as I said before, there are exceptions on this, but generally speaking, they're simpler, at least from the standpoint of organization, life form. Now, we mentioned the DNA, and we've seen in the test before that only few of you knew the DNA what it's for. So the DNA contains genetic information. It's a simple definition, but what does actually means? DNA contains the instruction to produce proteins. Proteins are a very special class of things inside cells that are a factory. They're very, very much building machines. So you need to imagine this like a factory, an engineering factory, where you have the plans to make everything inside the factory, and then the things to make can make other things, including the walls, the windows, the roads, the outside, and so on. The instruction is the DNA. The instruction on how to do things. When we say genetic information, we need instruction on how to make machines. The machines are the proteins. Very simple. Now, some proteins are complex machines, some other, they just hold the door open and so on. And then the proteins can make other things. They can modify the lipids, which are the walls, can modify sugars, they modify everything else. So in one way, when we speak about genomes, genetic content, genes in you, and your inheritance through childbirth and your ancestor and so on, we are just talking about the instruction on how to make things, mainly proteins. So how things work in biology, you have DNA, which can replicate itself. So you can make multiple copies of your DNA. That's what you pass out to other cells. It contains genes. You heard about genes before for sure. That's where the information is. Now, genes are the blueprint of how to make machines, but they never go around making machines. It's like I have one copy on how to build this place. I'm not going to give it to you to make the place. I'll make you photocopies, and then I'll give copies to everybody to build something. That midpoint, you may have heard or not, is called RNA. It's just how information is passed around. So DNA is where the information is. RNA is how information is given around. Now, not very important RNA in your case, DNA is important. And then the final part is the protein. The protein are assembled following the information that comes from DNA. DNA tells you what amino acid put. Protein are just a long list of different amino acids. DNA is just telling you which one to put next. It's like a giant Lego. You are puzzling things in a long chain. Proteins are who does the job, with simplification, because it's not as simple, but in the majority of the cases, explain as it is. DNA contains the information, protein do a job. Now, when you think about DNA in microbiology, you need to think of very simple things. Have you seen images of DNA of humans, the X, the Y, all the chromosomes line up? You might see them on TV, on series when they're searching for murder suspects and things like that. We contain a lot of DNA organized in several chromosomes that contain a lot of information around. Microbes are very simple. Generally, they have only one place where they have DNA, is a circle, a single circular molecule, where all the information are attached one next to each other. And they're fairly small in amount of information. So the average microbes contains about 2,000 or less genes, which means 2,000 instructions to make proteins, no more. That's it. And in spite of that, they can do a lot of damage as pathogens, as problems in underground hydrogen storage, but also a lot of good in the functioning of the planet. So generally speaking, DNA might be a lot in certain organisms, it is in higher organisms, but in microbes is relatively small in size, contains a relatively limited amount of genes, and is a single circular molecule. Proteins, on the other hand, which is who does the job in the cell, are a long list of amino acids next to each other, attached. When you attach amino acids next to each other, they interact due to van der Waals, hydrogen bonding. They have forces pulling them together. So they start to clump up in strange shape. Some of them are well structured, like they make helices going around, or they make folded area that are all flat together. And then they keep folding and folding and folding until you get something like the protein you see on the right. And you see there are ribbons going around, and you see there are flat areas with arrows moving around. This is just a linear structure, the folded, folded, and then attached to another linear structure, and you get globules in this way. Most proteins are globules, and they're globules not because of anything different than getting in the right place and doing the job. For example, some of them, they get two chemical compounds that they want to react, but they have an activation energy. What the protein does, gets the two of them, pushes them so close, then boom, they react together. So the shape of the protein is usually linked to the function that the protein has to do. Link things together, stretch things together, move things around. So protein do a lot of this job. Where is it? I lost it. Okay. Well, I wrote it here. So they're composed of amino acid. They can vary in complexity and size by order of magnitude. You have protein that are only 10 amino acids, protein that are hundreds or thousands of amino acids. They can be structural. They can just hold something in place. They can be sensorial. They sense something. For example, hydrogen in the environment. They can do signaling, that hydrogen is there and they send the signal somewhere. They can do storage. They can do transport. They can do physical, mechanical work. Stretching, attaching to two things and pulling them together. Stretching, attaching, and pulling them together. That's how your muscle works. Somebody stretching, attaching, and pulling all the time, every time you move a muscle. Or they can actually do chemistry. They can carry out reaction of different types. Hydrolyzation, lysis, redox, a lot of them. And some of these, the redox one, are what we're going to spend most of the time tomorrow discussing. So key message, DNA contains information. Who does the job are the proteins. And the proteins can do a lot of jobs, very different jobs out there. There's a special class of proteins. You also heard this name before, which is enzymes. That's a name you heard. That's a class of protein that catalyzes a chemical reaction. So the enzymes are not structural or sensing or mechanics. They catalyze chemical reaction. And enzymes are what concern the most your work in hydrogen, because you're looking for things that catalyze the reaction of hydrogen with other things. So that's where things are very, very important. And this is just to show you that there's also a scaling law between how big your DNA is and how many protein you can code. And you can see there's a pretty much a straight line, tapers off at the top, that's humans, at the top, eukaryotes and humans, because we have a lot of DNA that we don't know what it does. They used to call it junk DNA, but it's not junk, it's regulatory, we just don't know what it does. Microbes are very simple. First part of this graph, as long as your genome is, so many functions you have. It's actually very difficult, very easy to calculate. Every time we know how much is a genome, we know exactly how many functions, how many genes, how many coding things to expect. Straightforward, in this case. And we typically visualize it as this. You might have seen something like that, maybe not. This is one chromosome from one microorganism. Every short arrow you see is actually one gene, and they're coding for different things. And in an organism like this, there are maybe 5 or different genes to code for proteins that can catalyze a reaction with hydrogen. So different type of reaction with hydrogen. And you might hear biologists talking, oh, what type of hydrogenases contain? Is a single enzyme, multi-enzyme, things like that. Well, at least you know what they're talking about genes, what they're talking about enzymes, what they're talking about, and how they relate to each other. So just to recapitulate, There are a lot of things that might go on with prokaryotes. Most of them are additional or accessory. Oh, it's not a great quality of the figure, whatever. But you always find a chromosome, which is made of DNA, contains the information. You find the enzymes. There are the proteins that carry out chemical work. Then you have a membrane outside. Now there's a new name there, ribosomes, which is just the translating machine. This is what gets the DNA and makes the protein. There must be a middleman there, right? I'll give you the blueprint and you make a machine that does something. There's a machine that builds machine. It's called the ribosome. And that becomes relevant later when we talk about microbial diversity, because ribosomes are one of those things that is not just present in microbes, but is present in every life form on this planet. You contain millions of ribosomes in you that are making proteins all the time. And the color of your eyes, your nails, your hairs, whatever you have on you, your skin, is made of protein that are produced by this middleman, the ribosome, that is using genetic information and translate it into protein. And it's actually called translation, the process. Literally, that's how we call it in biology. Now, studying microbes, it's an interesting business because you don't see them, right? Or even if you see them, they're like a 1 micrometer bag. And they all look the same, or very often they look the same. There are a few morphologies, a few shapes you can see out there. But for the majority of the time, they look exactly the same. So we have 2 ways of studying microbes, 2 basic ways. The first one on the left is to grow them. You're going to try to grow the microbe in the lab and try to see what it does. What does it respires? What it grows on? Is it fast? Is it slow? Is it colored? Is it not? The other way is just sequencing their DNA. So you get their DNA, their basic genetic information, and you try to read it and see if it compares to anything you already know, which is what we do with murder scene. We get DNA from there, and we compare to a database to see if we already know that guy. We do the same with microbes. To make an analogy for you, to use a metaphor, it's like if you have a mixed bag of seeds, and you want to know what plant they are. Where either you plant them, and you wait for them to grow, and then you can recognize the plant, or you need to find another indirect way to classify them. Color, shape, size, dimension, and so on. That's exactly what we try to do with microbes. We do the top, growing them, and the bottom, using DNA to classify them. But it's no different than this. Now, this is fairly complicated in reality to do, because some time ago, we discovered that we're not very good at growing microbes. Actually, we are very, very bad at growing microbes. So in the 70s, relatively recent, in 85 actually, that's when the paper came out, but in the 70s, we had one of the biggest technological innovation in microbiology, which is called the epifluorescent microscope. So basically the idea is that you can shine UV light against something, and if you have a chemical that can fluoresce, gives you light back, okay? People designed chemicals that would fluoresce only if they are bound to DNA. So you dump the chemical in your sample, they enter inside the cell, they're very bad for human. Because of this, they're carcinogenic, they give you cancer. They go inside the cell, they bind to your DNA. If you shine UV light, they fluoresce back. By doing this, they will go at sea, get a liter of seawater, filter, color, count. And they will count 10 million microbes, 100 million microbes. And then they will do the culturing, grow them, and they will grow only like 600, 700, 800. And they will compare the numbers, and something is not right. So in 1985, these 2 scientists, Stanley and Konopka, published a paper called the Great Plate Count Anomaly. Every time I grow things on plate, I have a big anomaly of the counts compared to everything else. In 1985, which I was already 2 years old, probably you were not born, but it's recent for me, we discovered that there were a lot more microbes out there that we could grow. And we're actually very, very bad at it. So bad that in general, you can say that we can grow less than 1% of the type of microbes that are out there. So basically, going there, getting the seeds and seeding them is going to give you a very biased view of what microbes are in that place. And somebody did the exercise, and they went comparing for bacteria and archaea, two main group of prokaryotes, we'll see them later. They compared the percentage of sequences we get for just chunking DNA out of the environment and classifying it, compared to cultured things for different location. Human gut, hot spring, seawater, bioreactor, marine sediments, terrestrial subsurface. That's what's more interesting for you. And you can see, if you look at terrestrial subsurface, look up here, the amount of cultured microbes in terrestrial subsurface is less than 15%, with few exceptions in some locations. And if you want to know how many are not cultured yet, it's about 50% of average, but the bottom one is the scariest one, and we'll see why in a second. That's a taxonomic classification level called phyla or phylum. We'll see why it's very interesting later on. But basically, for the majority, for about 40% of every time we see DNA out there, we have zero cultured relative. Now, I know this sounds strange. What does it mean? I'll explain in a second, but we had zero representative members for an entire phyla. We'll see what a phyla is in a second. We'll go back to it. The idea is that we have basically not a single clue of what the majority of microbes do out there, and we have not a single clue of how to grow them. And actually, one of Guglielmo's projects is on trying to make this a little bit better using prediction, numerical prediction, and machine learning. So if you are out there, assume there's microbes, there's a lot of them, they're very small, we don't know what they do, and we cannot grow them. That's, if you use this basic assumption, you're gonna be right the majority of the time. Now, couple more slides and then we take a break. This is a figure published in 1967. Now, in 1967, this is how biologists will classify every living being on this planet. You have the plants, you have the fungi and the animals. You're all familiar with these three, right? At the bottom, you had protists, amoeba and plasmodium from malaria and things like that. And then at the very bottom, you had a domain they will call Monera, which will contain the prokaryotic non-nuclei single organism. You can see at the bottom Cyanophyta, which have the cyanobacteria, which are bacteria in the ocean that are able to do photosynthesis, they're primary producers, and then you have bacteria on the other side. Now, this is 1967. 10 years later, somebody had a great idea to use one of the pieces inside the middleman, the ribosome. You remember I told you ribosome translate DNA into proteins, and they're present in every life form of this planet. So one person had the great idea of use a piece of DNA of this middleman to try to compare things around. And he started to sequence and amplify and sequence and amplify and study this piece of DNA from the middleman everywhere, and realized quickly that things were not organized like this, 1967, but they were more organized like this. So on the right there, there's you and the plants and the fungi and the elephants and the dolphins and everything you see and around, including plants. And then there are two groups called bacteria and archaea, which are fundamentally different. We call them prokaryotes. We call them single cell organisms without nucleus inside, but they're more different between themselves than you are from a cricket, a fungi, a plant, or any other animal, insect out there. So basically, everything you see and you consider biodiversity usually is squashed to the right. And everything we don't see is actually two more domains of life. Now, well, somehow arbitrary, but you are up there. Now, this is the paper published in 1977. Now we've been going on sequencing and studying microbes everywhere. The actual tree of life is more like this one. Everything at the top is bacteria. At the bottom down here, you have this other domain of life, which is called archaea. And the green to the right are eukaryotes, right there at the bottom on the right. And in those eukaryotes down there, there's a single line. You see the tiny black line in which all life form, from yeast, baker's yeast, we used to make bread and beer, to elephant, fits inside in one of those single lines. So the entire diversity, you know, fits in that tiny line down there. Opisthoclonta. You, all other animals from yeast to elephants are down there. Now, if you think that you can squash the entire diversity of the planet that you know in a single line, and yet at the same level of distance, diversity, have all these other lines, it's pretty crazy, right? Now, we and all animals, from ascidians in the ocean to us, are a single phylum in classification, which is called chordata, means we have a spinal cord, okay? Chordata are a subset of things within a line down here, a tiny subset. Every other name you see up there is a phylum that contains as much diversity, if not more, that goes from ascidians to us, going through elephant and baker's yeast and so on. And every place where you see a red dot there is an entire phylum, so one of these giant group of things that are very diverse, for which we have zero information, no cultural information, we cannot grow anybody. It's like I told you, you go to a place, you have the entire diversity from ascidians to humans, and the only thing you see is a piece of DNA of one of these things. Can you imagine or reconstruct an elephant? Can you do the same with a butterfly? Can you reconstruct other things around? No, not really. But that's what we have for the majority of these things. And they can be more diverse than all the animals on this planet. So the diversity of microbes out there is stunning, incredible. They have the biggest diversity on this planet. When you speak about diversity, should not be plants and animals, should be microbes. And we know very little about it. So going back to key messages, they're a lot numerically, they're small, they're probably everywhere on this planet where you are within certain boundaries, we'll get to that, and they're incredibly diverse in ways you can only imagine. And tomorrow we'll see how diverse they are metabolically, which is what interests you as an expert of hydrogen. But for now, if you assume these 5 things, you are almost always correct. And as I told you before, we call them prokaryotes, which include the bacteria and the archaea, and consist of small unicellular organisms that are generally simple in structure. Now, prokaryote is not an official group like it is bacteria. Bacteria is an official taxonomy, an official classification group. Prokaryotes is more like a general term to refer to them, okay? So bacteria, archaea, and eukarya. You are eukaryotes, like the tiny algae in the ocean, like all the plants, like all the insects, all the animals, you are eukaryotes. Bacteria, archaea, different domain of life. And one more thing to tell before we take a break. You heard about the bacteria your entire life, probably, right? Every time there's a disease, it's bacteria. Lately, also viruses. But the majority of people, when we mention bacteria, they just think about pathogens. They think about disease. The reality is that we know about 16,000 of bacteria species. If you just take the list of the known species. If you include all the bacteria that we cannot culture, we probably have 150,000 types of bacteria out there, but we think it's actually more in the tens of millions of types of bacteria out there. Only 538 are pathogens. So out of hundreds of thousands or millions of bacteria that are present everywhere, we said 100 million to a billion for every liter of ocean or gram of soil. Only 538 are known pathogens to humans, and a little more if you think about pathogens of other animals and plants. So it's a very, 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 very tiny fraction. And of course, it's important because we do get sick, but the majority of bacteria, archaea, viruses are not dangerous for humans. They actually carry out environmental function. Okay? We take a few minute break, like 10-minute break. Riprendiamo. So we had a couple of questions before we started again. One was, what's the difference between genes and a genome? The genome is just the collection of all the genes of one organism. So you think about the genes are in the individual recipe inside that cookbook. The genome is the entire cookbook, how to make the entire organism. So in an organism, the chromosomes contains all the genes, and that will be referred to as the genome of the organism. While the single genes are the recipe to do a single piece. So how to do a chair, how to do a table, how to do a microphone, but the entire instruction to make the entire room, that's the genome. So genomes are the entire set of genes contained in one organism. And the other question was about the fact that when we bring samples in the lab to grow microbes, there must be a survival bias, right? What gets to the lab is what grows. To an extent, that's true. But it's also that some microbes don't like the environment we recreate them. You know, when a plant is growing in the forest, the soil is different, the shade of the plant is special, the air is special. There's a lot of very strange interaction with the environment. When you bring it back to your home, you try to grow it in your living room, doesn't always go well. The soil is not right, the amount of water is not right, the light is not right, the temperature is not right, and so on. The reality is that for microbes, we have no clue what is right or not, or we have it for a very small number of them. And I will say another Nobel Prize will go to the person that cracks how to cultivate uncultured microbes, because you open up manipulation of the gut microbiome, biotechnological application, you do a number of very interesting things. Any other question you want to have on the things we discussed so far?
Yeah, I'm wondering how they were able to upscale like the number of microbes in the I mean,
how they evolved, you mean?
It's approximate that number.
How we estimated the number. So different level of accuracy, right? But in one of these, we have thousands of samples collected from the oceans everywhere around the globe in hundreds of oceanographic cruises. And in each of those, we've been counting microbes. And so you can take different water masses and you can say, okay, in the Pacific, we have 1,000 samples and the average number of microbes is X. And so I can calculate the volume of the Pacific, and that will be my best estimate for microbes in the Pacific. And you repeat it for every ocean, and we did the same for soils, millions of samples of soil globally. We did the same with the subsurface because we've been drilling in hundreds of locations around the globe, both terrestrial and marine. And we've been filtering air and filtering ice and snow. And, you know, after you count a lot, of course, you have an error estimate around that number, but your estimates are pretty solid. And then every 10 to 20 years, somebody compiles all the new data and remakes a new, more accurate estimates. And yeah, we are within 1 to 2 order of magnitude error every time they adjust it. So we are not that far away from total numbers, if I understood well the question. So it's, you know, it's due to, your error is going to be due to sample representativity, number of sample, propagation of errors, and things like that. But overall, we are pretty confident that, you know, right now I can tell you that if you go out here and you take a scoop of the sand just on the beach, and it's average composition sand, is not special, not super thin, not super coarse, not super polluted, not, you know, extra clean and pristine, average sand. In 1 gram of sand, you have 10 to the 6 microbes. You can go ahead, take it and count it in 3 different ways. I'm not going to be that far away. Might be 5 to the 8, 10 to the 5, 6, 3, 10 to the 6, but we are in that ballpark because we've done thousands and thousands, hundreds of thousands of samples. We know what parameters controls this. Like if the sand is finer in granulometry, smaller grain size, your number of microbes goes up. If your sand is coarser, your number of microbes goes down. Why? Because they live attached to surfaces. The more coarse the sand, the rest of surface you have where to attach. but you're gonna have more pore water moving, right? And then you're gonna get more microbes in the fluids that they're able to move. And then it's not diffusion limited, your microbial system, but you have actual active transports for you modeling things. And with active transport, you increase connectivity. And there are all these things that are well known in the microbial world, and we can do very good models on how they influence each other. The bad part is that we spoke about genes and how they are the blueprint of remaking things, right? They have the information. And we know the middlemen, and we know the proteins. If you take the most well-studied microbes on this planet, which is called Escherichia coli, is the lab rat of every laboratory out there, not just microbiology laboratory, of every type of laboratory. It's been studied very intensively since the 70s. And now the way biologists study function is very interesting because we cannot draw. We have tried, there are a few papers that have done this, but drawing the minimal subset of things and make it work is very complex. It's not like you can design the minimal organism and make it and see if it works. Engineers do that. You do a simple circuit, it works, make it more complex, it works, you make it more and more complex. We have a very complex machine. And the only way we can interact with it is by cutting a piece at a time. We do that by canceling genes. We get an entire organism, we kill one gene, we cancel it, we make a copy, and we see what went wrong. It's like if I give you to adjust a radio, an engineer knows the principle of the radio, knows what to remove. In biology, we start to trial and error, cut things around to figure out what they do. By doing this, sometimes it's pretty obvious. You cut the antenna, you get no signal. You figure out the function. Sometimes you cut something, you're not sure. Nothing changed. Maybe it's changing the function you haven't tried yet, and so on. So with the organism like Escherichia coli, you can order an organism that contains every single one of his 4,000 genes canceled. You can order it. There's a company that makes them. So you can order a pre-cut version of your complex system and try to figure out, with that missing information, what is different. And you can even order 2 at a time. Every possible pairwise couple of things have been cut in this organism. You buy it, you order it, and even doing this from the 1970s to today, there's about 35% of the genome inside this organism, of the instruction. We have no idea what they do. You can cancel them, you see no difference. You can remove them, you have no idea what they're there, but they're used. They're even translated into proteins sometimes, but we have no idea. And that's the best studied organism. 35% of the information there, we have no clue why they're there. If you take the average environmental microbe, probably 70% of the information are there. You have no idea. It's like finding a cookbook, getting the project to do something. No clue what this is. Never seen it before. We don't even know if it's a machine for welding or for cutting. We have completely no idea what kind of machine. Sometimes we're not even sure it's a machine. We're not even sure it translates into a protein. The amount of information in DNA that is passed generation to generation, which means it's useful. Otherwise, it will get removed, right? We lost our tails. We don't need it. We lost connection between we don't have palm hands. We don't need them. Well, you don't use best adaptation. They're not under selective pressure. They might get lost. These things are in microbes, but we have no idea what they do. So that's our just direct ignorance on this. So very, very complicated and frustrating on one side, but with plenty of opportunities for great discoveries on the other side. Any other question? So microbes means small. There's a lot of them. They're tiny, around 1 micrometer on average in the environment. There's millions to billions of them almost everywhere if you are within certain boundaries, and we'll see what they are in a second. They're incredibly diverse, and they're composed of mainly 2 domains, which we call bacteria and archaea, which are fundamentally different from what we are, from animals are, what plants are, and everything else. And they contain a huge diversity within them. The other things to remember is that we really don't understand them very well. We cannot really grow them. And even when we sequence their DNA and we steal their blueprint, we can barely read 30 to 50% of what's written in there. Everything else is nonsense for us. So they hide a lot of what they do in the environment and what they might do. One thing that is interesting for engineers, physicists, and mathematicians is microbes are small, which means they experience the environment in a very different way than we do. What for us is a low viscosity fluid, for them is like a sticky, thick gel. They live life, everybody knows Reynolds numbers. It's a number used to describe behavior of fluidodynamics and viscosity. Well, they live their life a very low Reynolds number. For them, things are gel-like viscous. They don't swim in a soft water ocean. They move through a gelatinous space. They experience life in a very local scale, micrometer often, maximum millimeter scale. Diffusion things are very different, speeds, kinetics. So whatever you're imagining when you think about microbial interaction with your system, it's probably very different from what they experience. And microenvironment might be incredibly different from what you expect. An example is the surface oceans, where there's plenty of oxygen, not just in equilibrium with the atmosphere, but micro phytoplankton and algae are producing excess oxygen that goes into the atmosphere, right? There's more oxygen in the surface ocean during the day than there is in the atmosphere because it goes from ocean to atmosphere. And yet, if you sample the particles in the ocean and the seawater and you sequence the microbes, you do not only find microbes that are oxic, they're aerobic, they use oxygen, but you also find microbes that are adapted to stay in complete absence of oxygen. So how is possible? It's more than 21% gas space of oxygen in the ocean. We're producing oxygen with photosynthesis every day. How there can be there microbes that are adapted to no oxygen? They actually, if there is oxygen, they die. So why they're there? Well, in every tiny particle of dead organic matter, of sand floating, of fecal pellets of zooplankton and tiny things, the surface is in contact with the ocean, there's oxygen, but inside that tiny particle, which is probably less than a millimeter thick, half a millimeter, there's a network of tubes and porosities where oxygen is zero. So inside there are microbes that are perfectly adapted to anoxic condition. And it's like you have the entire planet in a single tiny particle that is less than half a millimeter big. You go from full oxygenated outside to completely reducing hydrogen and hydrogen sulfide rich inside. And you're going to find very different microbes on the surface and inside. So keep in mind that your experience of the world, the way you reason about the world, and the average condition you consider, they're very different from the tiny gradients that these things explore everywhere, right? Now, you can make simplification. In an entire reservoir, you can get, the bulk average production of H2S. But you need to keep in mind that that's a bulk average that represents areas where H2S is being produced by microbes very well, and areas where conditions maybe are more oxic or less reducing, and microbes are using H2S. So your bulk average is what it is, an average of very different environments. So keep that in mind. Life at low Reynolds numbers, completely different ball game than we are for everything around it. Now we'll mention briefly viruses. Now, they're not always relevant for the hydrogen story. We don't yet know their role in underground hydrogen reservoir, but we know they play a major role in every other ecosystem. They play a major role because we're used to think about viruses like something that infects human and give us the flu or COVID. But there are viruses that can infect every animal and plants out there, but also every bacteria and archaea out there. Bacteria and archaea have their own viruses. And lately we discovered the viruses that can infect other viruses. So you can bet that if there's an organism out there, it's got its own virus. And not one, probably multiple. So many people don't consider viruses to be alive because they cannot metabolize. What does it mean? It's similar to a USB drive. okay? Contains all the information needed to do something, but unless you plug it in a full computer that can read instruction and do something with it, it's completely useless. So viruses are a case, usually of proteins, with some DNA, usually, but not only, inside. But unless they plug into a full cell, there's nothing they can do. So they're very similar to a USB stick with the program in it. Unless you have a full computer, you're not running it. So many people call them biological entities, not organisms, because they cannot reproduce on their own. They cannot read their own DNA. They have no ribosome to translate them. They use the middleman of the machine, of the cell they're infecting. They're very, very abundant in the environment. On average, they're 10 times the amount of prokaryotes. So you count your prokaryotes, you can bet the virus are 10 times the prokaryotes. Everywhere we counted, no exception so far. I don't think anybody ever done virus in underground hydrogen storage. So that's Martina, that's something we can think about. They generally, a capsid, what we call a capsid, which is the USB drive made of protein, sometimes protein and lipids or protein and sugars, and then you have a piece of DNA inside. That's all there is in viruses. And we think 10 to the 31 on this planet. Now, when I told you before that microbes, prokaryotes, are 10 to the 29 to 10 to the 30, and viruses are 10 times more, so probably 10 to the 31, that's a number at any given moment. But if you want to know how fast viruses are infecting bacteria and prokaryotes, how fast they're destroying them, how fast microbes are multiplying, there's probably 10 to the 20 microbes produced every hour on this planet. So there's a huge turnover. Cells die and grow continuously. In an hour, there are hundreds of billions of millions of viruses that are produced and broken down all the time. So it's a very dynamic system. And because viruses can infect microbes and they destroy them, on this planet, in any moment, you can have an entire population of microbes going down, virus infecting them, population collapsing. And when a population collapses, a new population goes up until something happens. They get infected, they get destroyed, and they collapse again. And you get these cycles, very dynamic cycle of things going up and down. And sometimes when we do microbial incubation and we follow a population over time, we see a collapse and a change in abundance, and we are not sure. Might be viruses, might be other reason. So dynamic the system that we actually don't know what are the drivers there. But viruses play a huge role. I told you they contain mainly DNA. They're usually classified by what type of information they contain, but they may also contain RNA, so the working copy of information. There are complex viruses that contain DNA and make RNA and then back to DNA or vice versa. They have very complex life cycle going around. And this is technical, but you probably know it. There are viruses that infect your cell and start to multiply, like the flu or like COVID. Gets into your cell, starts to multiply, destroy your cell. You start to have problem in your lungs, you start to sneeze, you start to produce mucus, and so on. But there are also viruses that once they arrive in your cell, they enter, they don't reproduce. They stick their copy of DNA along your DNA and just leave it there for a while. And then when there's a reason of stress for the cell, the virus can sense the stress, is afraid the cell might die, pops out, makes copies, run away. Have you ever had a cold sore? Herpes cold sore in your face? Well, that's a virus that is sleeping inside your cell. It comes out when you're stressed, when you're immunocompromised, or otherwise. But you're living with a parasite virus in you all your entire life. Once you get it once, it just stays with you. We have plenty of examples like that outside. And they have very complex life cycle. But as you can see, they go in, reproduce and break, or in and sleep until they start to reproduce and break. Basic strategy for viruses. If I told you that bacteria have the highest biodiversity out there, viruses, they have the highest genetic diversity overall. Bacteria, we don't know 30 to 50%. Viruses, most of the time when we sequence them, 70 to 80 to 90% of the genes there. Not only we don't know what they do, but we never seen that genes anywhere else in the planet. And we keep sequencing, and every time we pick genes in the viral world, it's a completely new one. We have no way to relate it to anything else. And they have also the lowest number of cultured viruses, because to culture a virus, you need the host. You need the cell that you're going to infect and break. But because we can culture less than 1% of cells everywhere, or microbes everywhere, viruses are even less. So basically, some people call them the black box of biology. They do a lot, but we have no idea how, who, how often, and so on. Now, you have a question about viruses or bacteria? We'll go back to what I think are the basic information a few times. Any question meanwhile? No? So how common are extreme places on this planet? And what is exactly an extreme place? Because by any definition, living in an underground hydrogen storage site is not a common environment, right? You are underground, you're probably 60 to 80 degrees or more, weird pH, no oxygen, high hydrogen or CO2, hydrocarbons or other cushion gas, tiny pore space, no light, high salinity many times. So it's no way similar to the environment we are used to. The point is that when we think about extreme environment and extreme location, we always think about humans and what we like and what animals like us, mammals and or plants like. This is a very anthropocentric definition of this. If you think about our planet, the majority of our planet is different from what we like in one way or another extreme. So the majority of our planet's surface and subsurface is actually extreme. Think about the ocean. 70% of our planet is covered by ocean. Average depth is almost 4 kilometers. Average temperature of the deep ocean is 4 degrees. It's cold, it's dark, has a lot of pressure, and it's salty. For sure, not a place we can survive, neither the majority of animals and plants. And if you go into the subsurface, even worse, right? High pressure, tiny pore space, limited diffusivity of things, and yet we have microbes in the ocean everywhere. We have microbes in marine sediments and soils, and we have microbes in the crust. In the crust, we have microbes that extend probably 10 to 20 kilometers of depth, or at least anywhere we have less than 135 to 150 degrees. This is a small list of popular, of well-known, well-studied extreme environments on our planet. You see sea ice, permafrost, polar region, cold seeps, mud volcanoes, geothermal environment, hot spring, volcanic lake, desert, acid mine, drainage environment, deep sea anoxic brine, hydrothermal vent, serpentinizing environment. Those of you reading about natural hydrogen should know about serpentinization. Well, we have microbes that are perfectly adapted to pH 12 and high hydrogen in serpentinizing ecosystems. And then we have marine and continental subsurface and ophiolites, the counterpart of serpentinizing sites on land. And the marine and continental subsurface is where most of the action happens. But we talk a lot about porous media, but microbes like to live at very high salinities. And it's an open question, what's their role inside salt caverns, for example. And for sure, they love deep aquifers a lot. So the three most popular places where we are thinking about storing hydrogen, porous media, deep aquifers, and salt caverns, that's all within the domain of where life can be. So extreme places on this planet are common. Subsurface ecosystems are the second largest biome on this planet. Remember, 13% of global biomass is there. So what are the limits of life? When do you need to worry about life being there? Word of caution before we discuss this is that the limit of life is a moving target. In the 60s, we believed the upper temperature limit for life was 70 degrees. And then we moved it to 75. And then we moved it to 80. And then a very famous paper came out in Nature demonstrating that in hot springs, if you use radioactive phosphorus, to see incorporation of phosphorus into cells at 90 degrees, nothing grows. And therefore, they conclude in that very famous paper that nothing can grow above 90 degrees, as demonstrated by this famous experiment, probably because DNA becomes unstable and opens up, like we know. And so cells, they cannot deal with the opening of DNA, and they cannot survive. 5 years later, somebody isolated a microbe at 95 degrees. 7 years later, somebody isolated a microbe at 100 degrees. And you know what's the highest temperature we can grow microbes in the lab today? 122 degrees. How is that possible? At what temperature does water boil? You don't know what temperature water boils? Yeah, but under what conditions is 100 degrees? So pure water, atmospheric pressure. So if water is not pure or you're not at atmospheric pressure, temperature changes. So if you are a higher pressure, like underwater, few tens or thousands of meters, water boils at a much higher pressure, a much higher temperature, sorry. So you can still have liquid water for a longer time. And microbes are perfectly happy at the bottom of the ocean where water doesn't boil very high. Similarly, the coldest microbes can be grown in the lab right now. Take a polite guess. Coldest temperature. No, that's a little too much. We are around -20, -18 to -20. But you still need liquid water. How do you keep water liquid at that temperature? Pressure? No, pressure is actually salinity, solutes. You dissolve salts, your water doesn't freeze. It's a brine, super cold brine. Microbes are happy there. So this is a list from a paper we wrote a few years ago, where you have the current name of the most extreme microbes there. And we go from 122 degrees to -18, pH -0.05, all the way to 12.5 pH, salinity from 0 to saturation. So when you start to precipitate salt, microbes are still there and happy. You can increase pressure by a lot. So we grow microbes all the way to 121 megapascal, but microbes have been exposed to 2 GPa for you engineer and geologists, and they still survive. It's not a problem. You can squeeze them to 2 GPa. You can expose them outside the International Space Station to tiny amount of pressure, vacuum, essentially, and they still survive. They don't care. So the boundary range where life is huge, is enormous, right? So unless your water is boiling, you are above 100, let's say for safety, 135 to 150 degrees, because 122 is in the lab. But in the environment, we are not sure where the limit is. And we've been wrong before, multiple times. 95, 101, 103, 110, 113, 118, 119, 121, 122 right now. So who knows? So if you are anywhere where water does not boil, your water is liquid, it's not boiling, because thanks to pressure, for example. And your temperature is below, let's say, 135 or 140. Assume there are microbes there. That's a pretty safe assumption. Now, there is one more parameter that is important, which is space, physical pore space. We'll discuss that on Thursday morning, because that's relevant when you are in highly crystalline rock or salt. But, you know, to store hydrogen, we need space. So either we are in a porous media or we have a giant cavern there to host the hydrogen. So there is space. Space is not an issue. So the issue will be only temperature. In certain cases, chaotropic salts. Basalt cavern usually are allied, sodium chloride. So that's not a problem for microbes all the way to saturation, and temperature. So ultra deep porous reservoir are probably fine in terms of microbes because they're very hot. But we don't like reservoir to be too hot. Kinetics and dynamics change a lot. We probably don't want to be above 150 or 200 degrees. And therefore, you are generally within the domain of life. One exercise we did has been, so this is a type of plot we called polygon plot. It's showing you the range across multiple variables. In this case, you have salinity, pressure, pH, and temperature. of where you find liquid water on this planet. So this is not a state space type of plot. It's not like every point inside this plot represents a valid combination of this variable. It's just showing you the range, okay? Liquid water on this planet, this is the range. And you can see sometimes it's with high pressure, we can go high temperature and so on. Now, if you go and take every known life form on this planet, you map it, you get something like this. So life has adapted to use almost every possible salinities, a big range of temperature, not all of it. We are not sure why, or if that's accurate, or is going to change in the future. Almost the entire pH scale, and almost the entire range of pressures at which we can test things reliably. But people push the boundary going into GPA after this one. So the bottom line is, wherever you are working in your future, underground hydrogen or not, or whatever you're doing in the geological system, in this planet, anywhere, if you are within the boundary of known life, assume there are microbes, they're a lot, and they're very active. That's a safe assumption to start from. So I had a diagram that I developed some time ago that will tell you the if and when you should worry about microbes. So we left that on Thursday morning. It's like a flow chart for non-microbiologists. Should I worry? Yes, no, depending on the type of question. And I think that's what I like you to take away from this. It's a general understanding, very general understanding of what microbes are and they can do, and when and how you should worry about them in your job. So a few more slides of general things I need that are important. Biogeochemical cycles. I know that's a lot of disciplines in one word, but bio, geo, and chemistry. But this is how our planet works. So biogeochemical cycle are pathways by which a chemical substance, generally an element, under different forms. So same element that undergoes different changes as a molecule, sometimes different form, moves through the biotic and abiotic compartment of Earth. In other words, how it goes from soil to sediments to marine environment, back to the mantle, up into the atmosphere, into biology, back from biology to the soil, and so on. So any of this movement is part of what we call biogeochemistry. There are biogeochemical cycles well known for chemical elements, calcium, carbon, hydrogen, mercury, nitrogen, oxygen, phosphorus, and so on, as well as for water. But you can imagine and draw and study a biogeochemical cycle of anything, of plastics. What are the reaction and pathway through which plastics are transported and transformed in the environment? You can do it with a synthetic compound, LSD, PCB, anything you want. You can reconstruct what are the processes and the pathways that move a certain molecule or compound throughout different environment of this planet, okay? Usually, the biogeochemical cycle have a biotic side, the bio side, and an abiotic side, the geo side. Now, in some case, it's very simple to separate where the bio starts or ends and vice versa. In other case, it's not that easy to see where one stops and the other starts. So a good example is the carbon cycle. There are two versions of carbon cycle, what we call the surface cycle, the fast cycle, in which plants and biology, they take CO2 from the atmosphere, they make it into biomass, it goes around into trophic chain, we eat other things, until you die, you rotten in the soil, and you go back to be CO2 because microbes are degrading too. But that's the fast cycle on the surface. The slow cycle would be the same carbon ends up in the ocean, seafloor, gets buried under kilometers of sediments, becomes natural gas or something like that, and eventually the entire tectonic plate is pushed deep into the mantle and gets recycled by tectonic. It comes up millions years or billions years later in another part of the planet as new crust and new CO2 from volcanoes. We call that the slow cycle or the rock cycle. So when you are in the rock deep cycle in the mantle, you are sure there's no biotic component. It's purely abiotic. When you're talking about plants taking CO2 from the atmosphere with photosynthesis, you are sure talking only about biotic. There's no abiotic photosynthesis out there. But in a lot of instances of this planet, the two are not always clear-cut. They actually overlap a lot. And here I put two biogeochemical cycles designed by biologists in this, they're drawn by biologists. So they have molecules of that compound that are relevant for biology, connected by processes that are made by organisms. Left one is the nitrogen cycle. From N2 in the atmosphere, there are microbes that convert it into ammonia, then ammonia can be oxidized to nitrate and nitrite, and goes back, denitrified, and so on. This is a cycle that on the surface of the planet is mainly biological. There are very few abiotic contributions to this cycle, and we're talking about changing of redox state of this molecule. Transportation is mainly abiotic. Wind, rain, rivers, ocean currents, like transportation is mainly abiotic. But to convert things from one to the other is, I would say, 99% biological in this cycle. The only abiotic portion of the surface of the planet is ammonia made by lightning in the atmosphere. Hydrogen and two lightning, you make a tiny, tiny amount of ammonia in atmospheric chemistry. Now, if you look on the other side, on the other end, sulfur, which changes between two main states, sulfate, the very oxidized one in minerals and the ocean, there's a lot of sulfate in seawater, and H2S. The beautiful smell of rotten egg you will have on Wednesday afternoon as we visit our volcano. Those main two forms can be exchanged from one to the other by biology. But the oxidation of H2S in presence of oxygen is also abiotic. It's a fast reaction. It's a very reactive compound. So while one is going on, the other might also be going on, and you have no idea if what you're looking is biotic or abiotic, unless you focus on the process. And now we discuss this mainly tomorrow, but I want you to remember that biogeochemical cycle, they have biotic and abiotic processes, and they often overlap significantly. And of course, you can draw the same cycle in many different ways. This is sulfur again, left. Biologists are drawing the cycle between the main redox species, simplified, only 3 of them instead of 25 sub-redox possibilities. And you know, sulfur goes from -2 to +4. You can change all the way in, but these are the main microbes that do movement of sulfur. If you ask a geologist, he's going to look the other side. Everything you see here is this, and the biosphere. Everything else is transformation due to atmospheric chemistry, volcanic degassing, sediment and rock, leaching and erosion, and things like that. And of course, there is biology in rock weathering and in leaching, and there's biology in the ocean, but the clamp of that biology is that. On the other side, the biologists, they have very little care about all the abiotic reaction and the competition and the movement. Seems like all the sulfur is done in a single place by microbes. Reality lays at the interface between these two. So if you go out and you Google biogeochemical cycle of something, you should be able to recognize if you're seeing something drawn by an engineer, a biologist, a geologist, or whatnot, because their view is very much skewed to one or the other, okay? Absolutely. In every portion of that cycle here, so this one is connected with reservoir. Volcanic release to the atmosphere, back to land, from sediment and rocks, erosion goes from land to wastewater runoff into the ocean, ocean goes hydrological cycle, and deep rock cycle back to volcanoes, right? And then all this microbial stuff may happen on the volcano, on the rocks, on the sediment, on the land, on the wastewater, on the ocean, on sediment, going back into subduction. And of course, the different reaction microbes might do, they might not be everywhere at the same time, because if you're on the surface of soil, you have oxygen, you're going to tend to oxidize things. If you're deep under the sediment or deep in a porous reservoir, generally anoxic, there's oxygen, you might reduce oxidized compound the other way around. But it's a combination of the two, and they just have a very different granularity, right? We looking here at things that a single microbe does with one molecule. There we are looking for the exchange of things around. And instead of doing this, with my group, we've been drawing reaction, redox reaction that microbes do. We go back to those on Thursday. More like this. So this is thinking about one of the earliest versions we did. This is thinking about underground carbon storage, okay? CO2 at the center of it. And now, what microbes can do with CO2? Well, number one, they can make methane if we have hydrogen. And hydrogen in a reservoir might come from magmatism, metasomatism, radiolysis, hydrocarbons, or microbes, because there are fermenting microbes, number 7, that can produce hydrogen. But microbes can also use CO2 to break down methane and make CO2 again, number 2. But if you have CO2 there, you can also make acetate, number 5. If you have acetate, microbe can make methane from acetate, number 6. But CO2 can also come from hydrocarbon, because there are microbes that can break down hydrocarbons and make CO2. And of course, microbes can build things with CO2 and convert it into biomass, like plants do. We'll talk about that tomorrow. So you have 3, but then they can also respire it and respire out CO2 like you do. You break down sugar and you breathe out CO2. So that's number 4. And then you start to connect all these things, right? And you start to see how many reactions are possible theoretically. And then you should ask yourself, am I in the biological domain of life? Yes. Well, that's probably a tiny subset of all the things that microbe might be doing to my gas, CO2 in this case. So we'll go deeper into these on Thursday morning. And tomorrow we'll discuss what microbes can do metabolically. But the point is that this one is a portion of this that goes into the other one. So yes, they're a combination of the 2 things. One thing that people forget when we use textbook to study is that people that study carbon, they look for microbes that use carbon, like this. Oh, microbe can use CO2 and make methane, and then can use methane and make acetate, and then can use that to make biomass and so on. But if you're using CO2 to make methane and you are growing at the same time, you don't just need CO2. We are not made just of carbon. We're made of carbon, hydrogen, oxygen, nitrogen, phosphorus, iron, sulfur, and a number of other elements. A while ago, somebody called Redfield in 1930s went out in the ocean and started filtering seawater, million of points, and measuring the amount of carbon, nitrogen, phosphorus in every single point in the ocean. And he did this multiple times, different color, different ocean, Atlantic, Pacific, Indian. And he figured out there's a pretty consistent ratio, because proteins, sugar, lipids, DNA, doesn't matter if it's a microbe or is a plant or an elephant, they have the same chemical structure. So the amount of carbon to nitrogen to phosphorus that you need is more or less a set ratio. This one is called the Redfield ratio, in honor of the person that started this work. And it's more or less 106 carbon to 16 nitrogen to 1 phosphorus to 0.1 iron, and so on. You can make it as long as you want. So every time you're thinking about one cycle there, you need to think that a cell is a gateway where multiple cycles interact. So if you're thinking about utilization of hydrogen in your reservoir, sure, they can use it, but to use it, they also need the carbon. Where is the carbon coming from? And to grow with the carbon, they also need nitrogen. Where is the nitrogen coming from? In many locations on this planet, microbes could be a lot more abundant. They're not, because something is limiting their growth. It's like when you're growing a plant. Sometimes you need to add some fertilizer to balance out the elements. Otherwise, you can put all the light and the water you want. If there's no nitrogen or phosphorus in the soil, they cannot grow. Because a cell is a gateway where multiple cycles intersect. And it needs a fixed amount of different chemicals to grow. So very often, you have the potential for growing on a lot of hydrogen there, but there's zero available nitrogen anywhere. So microbes will not grow, even if they have a lot of food. They lack the building block to make the cell, because you need a set amount of elements. This is very, very important. Cycles intersect a lot. Very briefly, carbon cycle. most commonly studied one. As I said, fast cycle and slow cycle. The surface cycle is dominated by biology. What we call the slow or the rock cycle is dominated by geology and plate tectonic and recycling of different rocks and so on. They do intersect a lot more than we realize. And for your work, you are within the domain of the rock cycle, but you still with a lot of microbes acting on it. Well, you know this, or you can read it later. This is a depiction of carbon cycle from biogeochemists. So they don't think about the long-term geological cycle. This will be more like the surface carbon cycle in a changing ocean, okay, at sea. So you have the atmosphere, you have the ocean, you have the sea ice or brines at the top, and then you have the sediments at the bottom. And this is all exchanged. Some of them are abiotic. For example, calcium carbonate dissolution in equilibrium. That's completely abiotic. That's chemical equilibrium, dominated by pH and dominated by depth at which you are. Other portions like phytoplankton, grow and decay, and so on, are biological. But our integrated view on how these things interact becomes very, very important for you to model how microbes are interacting with your system, okay? Now, it's always good to start from a very simplified model, but you need to become realistic very quickly to make whatever assumption you're making on your system relevant. And being able to take a few of these cartoons and read them and see how they think things are connected and not, and then ask for help to draw one for whatever system you're working on, is probably the first and fundamental step in assessing if you have a microbial problem or not. Now, this one, as we said, plugs on this one, which is the slow carbon cycle. And everything you see here is actually happening only here. A tiny piece of seawater there somewhere. And this scheme here is not discussing what's happening in soil, is not discussing what's happening underground, is not discussing what's happening on rocks and so on. And you see how it plugs on the slow carbon cycle. So carbon, huge biological component on the surface, dominated by geology on the long term and deep, big anthropogenic imprint. Now, we're about to finish. I want to ask a question. What is the proof that we are responsible for the increase of CO2 on the atmosphere? Why do we think it's us? I mean, we say CO2 is increasing, right? And we have measurements that show there has been stably increasing for at least the past 75 years, since the Kneeling started measuring in Hawaii daily CO2 concentration over time. And we know from ice cores and things like that, the CO2 has been increasing for a long time now. But why do we say it's human-produced CO2 and not volcanoes?
There's a correlation between CO2 increase and...
Yeah, but that's not a proof, that's a correlation. So, well, in that case, it's not just the CO2 has been increasing, but the isotope signature of CO2 in the atmosphere has been changing. It's been becoming lighter and lighter and lighter, more depleted in delta 13 carbon. And that matches perfectly the amount of petrol and coal we burn. There is a very distinct isotopic signature. So, you know, there are interconnections there that we can see that tells us that, for example, there is a huge human footprint on the carbon cycle. There is not just by correlation, but we have much stronger proof there. It's not just about amounts, but carbon cycle, strongly biological component. Nitrogen cycle is a weird one because the majority of it is only biological. There's very little abiotic on the surface of the planet. Deep, we have no idea. There's a lot to be debated there, especially in volcanic area. Deep nitrogen cycle has been less investigated. Human activity also have a big impact on nitrogen cycle. Think about fertilizers we've been using everywhere. So we do have a huge input on that, still biological. And then last is sulfur cycle, just for your knowledge. It's very abiotic, dominated by volcano geothermal system, seawater. Life can be important locally, but it's generally not seen as a big biological cycle. Now, these three cycles are the most important one, and you will need to think about these three cycles when we discuss hydrogen, because hydrogen by itself is useless. You need to combine it to other things. And remember, cycles are interconnected, as we were saying before. Now, I put here an example of the phosphorus cycle connected to the carbon cycle, to the nitrogen cycle, and so on, just for you to review some of the basic and more direct connection we've been able to reconstruct. And I left a couple of readings here. I think the first one is probably the most interesting read you can do in general about what we discussed today. And the bottom one is a good view into one of the cycle, just to get in depth. Now, we are at the end. Questions? We condensated 2 months of microbiology in 2 hours.
But I have to check the salinity, the pressure, and the
Yep.
light. In that, you said the salinity is the maximum salinity that you find in the sea, or?
No. That's 35%. That's 10 times C. And so there are several locations in which we found microbes at 10 times the concentration of seawater. One of the most fascinating ones are called deep hypersaline anoxic basin. They're basically giant anoxic lakes in the bottom of the Mediterranean, the leftover from the Messinian salinity crisis. The thalassic one, so-called thalassic one, which means they have the same composition of seawater, they're 5 to 10 times more concentrated than seawater. There's plenty of happy microbes living there. Oh yeah, all over the place. And any salt pan where you are producing salt, you have microbes all over the place. And they're actually bright red in color.
When we are able in the future to build to the mantle, do you think we'll find microbes there?
So that's a question that goes back to what we think is the highest temperature and what we think is actually needed space. Because as you go deeper and deeper in drilling into crystalline bedrocks, your pore space goes down to 0. And if there's 0 pore space and no connectivity, you have no space for microbes to be. Probably they might be concentrated around fractures where there is circulation of deep fluids. And so that's possible. The microbes are not in the rock per se, but they're circulating within deep or ultra deep aquifers. And we have proof that that's possible from ultra deep diamonds and gold mines in Canada and South Africa, where people have found microbes in pockets of brines within crystalline bedrock at 4.5 kilometers depth. Now, when you say we're going to drill into the mantle, that's a different story. Not just when, but what do you think the boundary to the mantle is? Because we have this moho discontinuity, but we're not sure how it reflects into the geochemical or petrological discontinuities. So I'm actually part of one project that has been drilling in an area of the Alps where we have a gravimetric anomaly that suggests that the moho is very, very shallow, only 3.5 kilometers. And we did 2 pilot holes down to 1 kilometer. Guillermo also worked on one of the drill sites, Martina as well. And probably in 2 to 3 years' time, we're going to move into phase 2 and try to drill down. Now, in the 2 areas where we drilled, where we found granulite, metapelites, and things that are pretty crystalline, I don't think we're going to see microbes in the rocks. I think we're going to find them only in the waters that are flushing some of the fractures. If it's a cold mantle transition, which I don't think it exists, but we might find surprises there. I mean, there are work that suggest that in cold subduction zones like the Marianas, you might have microbes alive up to 20 to 25 kilometers depth in the slab going deep into the mantle. So theoretically possible. Depends on condition. Any other question? Yes.
The last scale for condition for this just for insight, what are the frequency control cycles that you mentioned earlier?
Can you repeat the second part? What are the
Frequency of the cycles, like how fast those cycles are?
cycles? You mean?
Like both in sulfur, nitrogen, or carbon?
Well, that depends on the activity of your microbes. So we have a big problem in microbiology, we'll discuss it tomorrow, is that we cannot really measure rates of chemical reaction from microbes in situ. The only thing we can do is to bring back the sample and measure that reaction carried out by microbes in the lab. But to do it, usually we are changing condition and stimulating the microbes, which means what we get is a rate, and we're not sure how does it relates to the bottom one, you know, the deep one. It will be faster, slower, and so it's difficult. Right now, people will put a lot of money into new ways to measure chemical rates by microbes inside the reservoir. Now, just to give you an example, microbes might be as fast at consuming certain things, they can completely change the redox state of very large reservoir in a matter of days. So technically speaking, some of the cycle might cycle a lot and very often, might be very fast. But that's only theoretical because nobody ever did it there. Microbes can kill your giant aquarium in 2 to 3 days if you're not filtering and aerating and moving things around. Microbes made this entire planet toxic in a few tens of million years. That's a big one. And, you know, there are very fast cycles out there controlled by microbes. Think that microbial activity in the winter in certain organic rich feature can actually generate self-combustion of whatever you are looking at, because they produce so much heat from the metabolic work they're doing that, you know, you overheat when you work out, they overheat enough, they're in wood chips, they actually generate fire on their own. They smoke in the winter. So potentially very fast. They can turn your fruit juice into wine in less than a week. The problem is that practically we never did measurements down there. We don't know how to do them. So we can only come up with theoretical rates right now, and we don't know how they compare to in situ rates. So generally people will speak of potential for microbial sulfate reduction to H2S in hydrogen storage site, and then tell you that under the tested condition in the laboratories, these were the rates. But the two things are completely disconnected. And the biggest gap is for microbiologists to find the reliable ways to assess these parameters to be able to provide to the modelers a reliable estimate. And that's why some people, not us, get millions of dollars from governments to do this. Any other question? Well, I'll see you tomorrow morning, and we'll talk about what metabolism is and what microbes can do, and how thermodynamics and kinetics control biotic versus abiotic reaction in whatever system you are working on. Thank you.